Now let's focus on the movement into and out of cells segment of this specific chapter. Now, what are the different ways in which movement can actually happen between cells? The first method is something that you have known pretty well for a long time, and that is diffusion. Something new that is possibly going to be introduced to you along with diffusion would be we'll be talking about a specific improved type of diffusion known as facilitated diffusion. You'll see what that exactly is. Then, number two, we have to talk about osmosis, something you have also known. Active transport, same spec. Transport. And then we have something interesting and quite new known as bulk transport. And bulk transport actually comes in two types. Number one, we have something known as endocytosis. And number two, we have something known as exocytosis. As for the meaning of any of these, don't worry, a detailed account will be provided as the chapter continues. Let us start off with number one. Let's talk about diffusion and facilitated diffusion as well. Oops, that is not thick enough. Diffusion and FAC, facilitated diffusion. Start off with a nice little color. I think I'll use a shade of blue. Yep, that tracks. I will first of all start off by giving you a formal definition of what diffusion is and how you are expected to define diffusion in the examination. I'll type this out. Diffusion is defined as the net movement of molecules from a region of higher concentration. Now, you have known this definition since your O-levels and or IGCSEs, but this is going to be a little bit of an improved definition. Nothing's going to change. It's the exact same definition. In any given point, at any given point, if a question states define diffusion, this will be the definition you're going to give for a 2-mark question, 3-mark question, 4-mark question. In any case, you will get the full marks if you follow through this definition completely. From a region of higher concentration to a region of lower concentration. We always know things go from a high to low concentration. So low concentration down the concentration gradient passively. What do you mean by passively? Passively means we're not going to be using any form of energy. Passively as a result of the random motion. There are no specific driving forces for diffusion. There are no specific laws that are going to govern diffusion. It's just a result of the random motion of the particles that are present. Okay, then let's talk about the different factors that can affect diffusion. Factors affecting the rate of diffusion. The first factor, and perhaps the most obvious one, is temperature. When temperature is increased, particles have higher kinetic energy, therefore they move faster. If particles are obviously moving faster, the rate of diffusion, the rate of movement of those particles are pretty high. Number two, concentration. Gradient. What does that mean? Now, if you have a steeper concentration gradient, so I'm going to denote it as being a steeper gradient, you will have a faster diffusion rate. What does that mean? Take this for an example. Now, if you have two scenarios, let's denote left side is scenario A and scenario B over here. Now, if this is the boundary, let's say I have 1, 2, 3, 4, 5 and one molecule on this side. So what's the direction of diffusion? Things will go from here to here. Duh, right? High concentration to a region of lower concentration. Now, if I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and just one here, still the concentration gradient is the same, right? We're going from a high to a low region. But do you see the steepness of the concentration gradient? Here it's 5:1. 5:1. Here it's around, how much did I draw? 4, 5, 6, 7, 8, 9, 10, 11, 12. It's 12:1. I'm so sorry if I counted wrong, but it's 12:1 versus 5:1. What's going to move faster? Scenario B, things are going to diffuse or move down faster. Why? Because we have a steeper concentration gradient. That's the idea. Let's get rid of all of that. The third factor would be the surface area. Surface area available for particles to move. So obviously, if you have a higher surface area, more molecules pass through. Still, all of this is IGCSE knowledge, so the rate is ultimately going to be increased. It's going to be ultimately increased. The fourth and final factor would be the size of the diffusing molecules. Size of the diffusing molecules. Lower the molecule size, higher the rate. Okay. If we have a lower molecular size, we have a greater rate. Okay, very simple. Those are the four factors that are going to affect diffusion. Now, what about facilitated diffusion? Now, this was the formal definition for diffusion. So let's go through with a formal definition for facilitated diffusion, which I will also type out. DEF for definition. Like I've said 8,000 times, if at any given point you have a DEF bracket, that means that is a formal definition and you should memorize it for the usage within any examination paper. Right, how would I define facilitated diffusion? Facilitated diffusion is actually a form of diffusion, obviously, as the name suggests. The thing is, this involves special transport proteins. Those transport proteins are going to help substances that normally cannot pass through the cell membrane to pass through the cell membrane. That's the importance of facilitated diffusion. Now, what do I mean by things that cannot pass through the cellular membrane? If you remember the structure of the cell membrane, going back all the way to the first part of the thing, we specifically discussed how hydrophilic, if you have no idea of what I'm talking about, do go back to the previous videos. You can get a better clear understanding of what I'm talking about, but I'll just give you a basic short summary right now. If the molecule is hydrophilic, if the molecule is hydrophobic, meaning if it does not like water, it can straight away cross through. It's not a problem. Why and how, like I said, you'll get more information from the previous specs, but just telling you hydrophobic molecules can pass through. But if you are a hydrophilic, water-loving molecule, you just cannot pass through the cellular membrane. It's not possible. You have to go through special channel proteins. If you are doing that, the process that you are undergoing is known as facilitated diffusion. Right? So just as a small summary, I'll give you the non-passable molecules for some examples. Samples for non-passable molecules. Polar molecules. That's the first type. These are non-passable molecules, alright? Polar molecules. And then charged ions. These are just two examples. Okay? These things just cannot cross the cellular membrane straight away. Okay? They need some form of what? They need some form of special transport protein. They need some special transport proteins to help them pass through. Okay? Under the context and concept of transport proteins, I should mention that there are two highly specific transport proteins. Handwriting is going haywire. The first type of proteins that we have, we denote them as being called channel proteins. They are known as channel proteins. A small information segment about channel proteins. Channel proteins are simply water-filled pores. That's why hydrophilic molecules, like polar molecules, like charged ions, can freely pass through channel proteins. I showed you a channel protein before from the membrane structure. See, this is basically a water-filled pore. If you didn't have context here, this is just a water-filled pore. If you want a 3D structure and a better understanding, Google's always your friend. So give it a small search of a 3D structure and you'll have even a better idea of what a channel protein is and what a channel protein does. Now then, channel proteins allow charged substances. What do you mean by a charged substance? A charged substance is an ion. So for example, Ca2 plus, Na plus, Cl minus C. The charged stuff. That's what we talk about. To pass through. And the last point I have to mention about them is that they are gated. We typically refer to them as being gated ion channels or gated channel proteins, meaning their opening or closing is heavily tightly controlled. Heavily tightly controlled. That's the first type. The second type, now I'll just simply draw a channel protein action. And boom. This is the phospholipid bilayer. These proteins are of course found in the phospholipid bilayer, like this. And this is a water-filled pore. So I'll denote the water like that, and things can freely pass through like that. The second kind of, now we said we have two highly specific types of transport proteins, and we discussed the first one. We call them channel proteins. The second type is known as I kind of gave it away previously, is known as a carrier protein. Okay, now the key thing about, now you see channel proteins are fixed. No? They're fixed in shape. They're literally just the channel. But when it comes to carrier proteins, the most distinguishing feature is that they can switch between two shapes. They can switch between two shapes. Okay, that's the important thing. They can switch between two shapes. How would that look like? Let me show a live example. If this is my cell membrane and this is my channel protein, something like this can happen. This is its initial shape. I'll call it shape A. Now, if this is my molecule that has to go through, this is going to go through, it will come and fit onto the channel protein. I'm sorry, carrier protein. I'm wrong, not channel, carrier protein. And then this is going to open scenario B or shape B. Let's get rid of that. Shape B. Okay. Now this carrier protein is going to flip its shape, meaning it's going to change like this. Observe very closely what's happening. It is going to shoot out like this. When that happens, what's going to happen to this molecule? It will be propelled inside. It will be propelled inside into the cell membrane. Okay, shape A followed by shape B. That's the important thing. Channel protein does straight away one shape. They just allow things to pass through. Carrier proteins can switch between two shapes. What do they have in common? What they have in common is channel and carrier proteins all support the function of facilitated diffusion. Okay, that's the key idea you have to get. That's all about diffusion that you have to know. Now, the second type of transport you have to be aware of is going to be, again, something you've already known, but of course with improved insights. That's going to be osmosis. Let me double line that with another blue. OSMOSIS. Same deal, formal definition. How would you define osmosis? Osmosis is the movement of water molecules. We specifically talk about water when it comes to osmosis from a region of higher concentration. Is that right? No. If you say higher concentration, my god, why did I move to organic chemistry? 1 second. Okay. Plot twist, organic chemistry. Never mind. From region of higher concentration. Is that right? Can we talk about concentration when it comes to water? I mean, yes, very strictly or unstrictly speaking, yes, you can talk about concentration, but we don't usually talk about concentration when it comes to water. We say water potential. We don't really talk about concentration with water. With solutes, we talk about concentration, and that's perfectly admirable, that's perfectly understandable. But when it comes to water specifically, we don't really use the term concentration, we say water potential. So this is when water is moving from a region of higher water potential to a region of lower water potential down the water potential gradient, just like the concentration gradient. Here's the kicker when it comes to osmosis. We have to go through a semi or partially permeable membrane. Okay, we have to go through a partially permeable membrane. For example, a cell membrane. A cell membrane is partially permeable. That we established. That's the idea. Now, the water potential of a solution is described as the tendency of this water to move out of a solution. To move out of a solution. Okay. A very important metric you have to keep in mind is the water potential of perfectly pure water is zero. The units are actually kilopascals. You don't have to wonder, you don't have to worry how this just came to be. But just so you know, at atmospheric pressure and temperature, the water potential of pure water is zero. Okay, the tendency of water to move out of the solution when it comes to pure water is zero. Okay, water potential is shown by this special symbol that looks almost like a trident. Something like this. Psi W, that's what we call it. This is what represents water potential. It almost looks like a trident, and we have this W to denote water. It's almost like the potential of water. More on water potential and why all psi W values are negative later on in the spec. For now, that's all you have to know about osmosis. Very simple, very straightforward. Now let's move on to the third type, active transport. This is also nothing new. Same deal. Let's define active transport before we do anything else. Active transport is the movement of molecules or ions from a region of, here's the difference and don't mess this up. Active transport, now we're going in the reverse direction. Usually in nature what happens is something moves from a high to low level. High concentration to low concentration. High water potential to low water potential. Here we're going the opposite way. The movement of molecules or ions from a region of lower concentration to a region of higher concentration. We're going in the opposite direction. And therefore, we have to specifically say we're going against the concentration gradient. We're going against the concentration gradient. For us to be able to do that, we need energy in the form of ATP. In the form of ATP, which we obtain from cellular respiration. Three key things to keep in mind. We're going from lower to a higher concentration. That means the second thing, we're going against the concentration gradient. Third thing, we need ATP. That's the form of energy, and that is produced by cellular respiration. Okay. The important distinction I'm going to give to you right now is active transport also uses carrier proteins. Okay, also uses carrier proteins, right? But these carrier proteins are different from the facilitated diffusion ones. So it's a diffusion ones. Okay, why? As energy is being used. For facilitated diffusion, we had carrier proteins, but did we use any form of energy? No. Diffusion is a very passive form of transport. Passive means we don't want energy. But this here, we're talking about active transport. We clearly need energy. One of the best examples of active transport, one of the best examples for active transporter molecules that I can give you is the This is going to be a very common thing occurring throughout biology, the sodium potassium pump, typically shortly written as NaK pump, Na plus K plus pump. Simply, this is what is happening. We have something like this. That's the protein, that's the transporter protein, and this is kept between the bilayer like this. And this is going to transport sodium and potassium like this. 3 sodium comes in, and at the same time 2 potassiums are made to leave. More about sodium potassium pumps later. You don't have to know that much about them. With that discussion of the sodium potassium pump, we can move on to the final type of transport, and this is going to be the most new type of transport for you, bulk transport. Like the name suggests, bulk transport is induced when we have a lot of things to transport. Bulk transport mainly occurs in two types, endocytosis and exocytosis. I'll tell you the differences in a bit. Now, let's start off with a few introductory points because this is a new part to you. We talked about 3 methods before. Like I said, we have been moving individual molecules. Now, if we are using bulk forms of transport, large molecules like proteins or parts of cells can be transported. Okay, we don't have to rely on just individual molecules. We can transport an entire fleet. So then how and why is this so special? Let me pick a color. I think purple's good. Right, endocytosis. Let me first tell you about endocytosis. Just think of exothermic and endothermic basically. Cytosis means we're taking things into cells. On the same front, exocytosis means we're taking things out of cells. Okay? The key thing to understand is both of them they require energy. Therefore, they are deemed as active forms of transport, known as active types of transport, active forms of transport. Let's begin with endocytosis. I'll just go endo dot endocytosis. Endo means in. Okay, endo means in. Definition: endocytosis can be defined as the process by which the cellular membrane engulfs material. Cellular membrane engulfs material, forming a sac around it. When this happens, we call this a endocytic vacuole. Vacuole. A small demonstration. Let's look here. Let's say I have some particles, and here's the cell. Now what this cell is going to do is it's going to recognize, oh, I have to engulf these particles. So I should do something like this. So it's going to go close by to the particles and it's going to try to engulf these particles. So something like this forms. This cell is going to form this thing. This thing is known as an endocytic vacuole. See, it's literally engulfing the thing. So afterwards, it would break off and we'd have the endocytic vacuole like this. The cell membrane would have regenerated, not an issue. Inside, we'll have the endocytic vacuole with that uptaken particle, which is this thing. That's the idea of how cells engulf materials. Right? Now, when it comes to endocytosis, we also have two forms, or let's call it two types. The first type is phagocytosis. So under your syllabus, you have to know of these two types when it comes to endocytosis. The first type is phagocytosis, which is the bulk intake of solid materials. Okay, cells specialized in this, that's where specialized in this are known as phagocytic cells. Known as phagocytic cells. And what's the second type of endocytosis? That would be, it has a little bit of a funny name, but pinocytosis. It would be pinocytosis. This would be the bulk intake of liquid molecules. A very simple, clear, distinct definition we have. If it's a solid, it's phago. If it's a liquid, it's pino. That's all. That is very simple. Now you will be actually learning about phagocytes a lot under immunity. Under immunobiology, you'll be focusing on phagocytes a lot. So if some foreign thing enters our body, phagocytes go and engulf that. Like a bacterium or a pathogen, if it enters, our immune cells our phagocytic cells will go and engulf them through endocytosis, through phagocytosis. More on that under immunity. Onwards to the exo part. Exo means out, just like exothermic. Exocytosis is the process by which materials are transported, this time out by again secretory vesicles formed by this time the Golgi apparatus. If you go back to spread 1 of this particular course, AS Biology, you learn everything you need to know about Golgi apparatus and how they facilitate exocytosis and the removal of materials. With all of that complete, that is the end of this specific spread. Went back a little too much. Right here. Movement into and out of cells. We discussed the major 4 ways in which cells can move substances both into and out of their respective surfaces.